Okay, it's 12 o'clock, so it's time to start the last installment of this semester series of Polish NMR seminars. Today we'll have two talks, student talk and main talk, but before that, as usual, I would like to advertise best student question competition. So I encourage all students to ask questions. You can win a nice set of Polish Chemical Society gadgets. You can ask Adam Kubrak, who is in the audience, that those are real. And due to the small amount of questions this semester, any question asked today will be transferred for the competition for next semester. So with that, I would like first to introduce Maria Rutkowska, the youngest ever speaker on our seminars. She's just finishing her bachelor of studies, but was brave enough to give us a talk. So her topic will be insight into the competition between reisomerization, hydrogenation, hyperpolarization of exemplary pyridine-based photoswitchable molecules. So Maria, I'm stopping sharing and the screen is yours. Stop share. Okay.
Hello, my name is Maria Rutkowska. I'm a bachelor student of nuclear chemistry and radiopharmaceuticals at University of Warsaw at Faculty of Chemistry. And I also do my thesis at Institute of Physical Chemistry, Polish Academy of Science at Applied NMR Group. And today I will be talking about insight into the competition between reisomerization, hydrogenation, and hyperpolarization of exemplary pyridine-based photoswitchable molecules. To start off, I will be talking about how do we induce hyperpolarization. But before we jump into the hyperpolarization, I want to talk about parahydrogen and how we utilize it to hyperpolarization. So before we talk about parahydrogen, let's say what is a parahydrogen. And in molecular hydrogen, there's two spin isomers of hydrogen. There is orthohydrogen and parahydrogen. In orthohydrogen, the spins are parallel to each other and it is a triplet state. And in parahydrogen, the spins are anti-parallel to each other and it is a singlet state. Because of that, we can induce hyperpolarization to our main problem of NMR, which is low sensitivity. And as you can see on the diagram on the upper left of this slide, in normal equilibrium, the spins are with our point of hyperpolarization is to drastically disrupt this equilibrium that is normal in room temperature, and with hyperpolarization this equilibrium is changed, so more spin can give us a signal that is measurable in NMR spectra. And we obtain parahydrogen by simply lowering the temperature of our gas. To enrich hydrogen in parahydrogen, we use liquid nitrogen that is around 77 kelvins. And at this temperature, we have around 50% of parahydrogen and around 50% of orthohydrogen in comparison to 75% of orthohydrogen in 300 kelvins. So coming to hyperpolarization, in this work, we decided to use SABRE, and SABRE is signal amplification by reversible exchange. It's popular technique used in hyperpolarization that utilized parahydrogen. However, the most important part of SABRE is utilizing the catalyst. And in my work, I use iridium IMS catalyst. As you can see on the screen, it has a iridium complex center to which parahydrogen is binding in 2 positions, and it also has 3 positions in which ligands can be coordinated, and those ligands are what we are working on. As you can see, the hyperpolarization coming from para-hydrogen is coming to the ligand that we're working on through the iridium center. In my work, I also used photoswitch molecules, and those photoswitch molecules can be transferred from E to Z isomer using light. In this work, I used phenyldiazenyl pyridine that can be transformed from E to Z isomer using light. And in this work, I focused mainly on the hyperpolarization of this molecule, but also in how its reisomerization and hydrogenation, but we'll talk about it in a minute. And hyperpolarization of E isomer is pretty easy. It's really good in hyperpolarization. It's hyperpolarization really easily. It's stable. It doesn't change even after many cycles for what we know now. This topic needs to be also later more. We need to put some little bit more work into that, but for what we know now, it's stable. It isn't hydrogenation. And of course, it's something that it doesn't transform to the isomer on its own without the induced light. However, what is interesting is that the isomer in hyperpolarization, of course, it's reisomerizing into the E isomer. As you can see on the NMR stack spectra on the left side on the screen, it's slowly transforming to trans isomer, onto the E isomer. However, what's also interesting, there are some new peaks showing up on the spectra. And this was also the point in my work, to get to know what is the thing that we're obtaining during this hyperpolarization. And we came to a conclusion that is hydrogenation. So this Z isomer is hydrogenating and reisomerizing to E isomer at the same time. I would really like to thank you all for listening. I would also really thank to my thesis supervisor, Dr. Tomasz Atajczyk, PhD, and Piotr Garbacz, PhD, and to all my colleagues at the Institute. Thank you very much.
Thank you, Maria, for your talk. And we are open for the discussion. If you want to ask questions, please turn on your microphone and, if possible, camera and ask the question. Do we have any questions? Maybe the supervisors would like to know a little bit about the project.
Oh, I think so. I know everything.
Okay, so
okay, thank you very much.
In that case, thank you very much, Maria, for your talk. And let me introduce our next speaker. So it's my great pleasure to introduce now Dr. Katarzyna Malec from Wroclaw Medical University. I invited her mostly because she got the prize as the best PhD thesis from Prime Minister of Poland this year. So this is how I found out about her. So I would like to introduce her talk, the application of NMR spectroscopy in drug formulation technology. So thank you very much, Katarzyna, for joining us. I'm switching off the sharing and you can share your screen. Okay, everything works, so you can start.
Good afternoon, everyone. Thank you very much for the invitation and also a very kind introduction. I work at Wroclaw Medical University at Department of Drug Form Technology. And today I would like to present some examples of the application of NMR spectroscopy in drug formulation technology. At our department, we develop several drug formulations, mainly colloidal materials, semi-solid forms, and solid forms. We try to optimize either the composition of the formulation or technological parameters of the processes that we are using, or the synthesis pathway. All of this to get the product with specific characteristics, such as physicochemical properties, release profile, or to adjust the formulation to the specific route of administration. So at the beginning, I would like to list 5 areas of research that I'm conducting in which NMR is applied to characterize drug carriers. The first one is molecular level interaction in drug delivery systems. Materials used in this study are micelles and liposomes that form supramolecular assemblies. Micelles are small nano-sized drug delivery systems built by surface active agents, and recently the most common are polymeric micelles. While liposomes are small vesicles built by different lipids that form lipid bilayer, and they have aqueous vesicle inside. The other area is drug formulation stability over time, using the example of nanoemulsions. In this case, nanoemulsions are a multi-phase drug formulation which contain water phase and oil phase, and they are stabilized by the mixture of surfactant and co-surfactant. And in this case, the stability, long-term stability, is the main concern upon preparation. The other area is molecular level characterization of the external surface of functionalized nanoparticles, such as mesoporous silica nanoparticles. These particles have the functional group attached to the surface that enable the targeted delivery or they increase the colloidal stability of these particles because they tend to aggregate when the suspension of MSN is created. So the challenge in this case is the analysis of this external surface that stems from surface area because internal area of the porous materials is much larger than the external one. Further field is the impact of the manufacturing process on the product. For example, liposomes, after preparation of the liposomal suspension, to get uniform suspension, we use either extrusion or sonication process. Sonication is very common at the moment. However, it might destroy the structure, the three-dimensional structure of the liposomes, and NMR might be applied to look at these changes. Another example is UV-cured gels manufactured by semi-solid extrusion. In this case, NMR was used to confirm the photopolymerization process that was the effect of solidification of the gel into a solid form. And the last area is the use of NMR to explain the mechanism of action of the formulations in biological systems. In case of resistant Candida strains, we could observe the potential inhibition of the overexpressed efflux pumps in the yeast by fluconazole-loaded micelles after application NMR. In we could also observe the changes in wall structure and membrane structure after interaction, after application of essential oil-loaded emulsions on methicillin-resistant Staphylococcus aureus. That the aim of the project was to explain the mechanism of action of those essential oils that might have antibacterial properties. Depending on the project, we use solution state NMR, high resolution mass NMR, or solid state NMR. And today I would like to focus on two projects that involved solution state NMR. So the first one is molecular level interaction in micellar systems that were investigated by a novel differential epitope mapping by saturation transfer difference NMR method. And as short introduction, micellar assemblies have many advantages as drug delivery systems. They might increase the solubility of purely soluble active pharmaceutical ingredients, APIs. They might modify the drug release. They might be used as for targeted drug delivery, and they can be applied to variable routes of administration. So at specific concentration called critical micellization concentration, CMC, surfactant monomers assemble into the micelles, and there is constant exchange between monomers and aggregates in such formulation, so the structure is quite dynamic. And after addition of the drug, it might be localized in variable sites, so either in the core, at the interface of hydrophilic hydrophobic domains, or at the surface of the micelles. So in this project, systems under investigation comprised 4 different surfactants that belong to different type of classes of surfactants, such as non-ionic block polymer, ionic surfactant, and non-ionic surfactant. They differed from each other with molecular weight and CMC. And as the model drug, we used 2 drugs that differ from each other with polarity and solubility in water. So fluconazole that has more hydrophilic properties, and indomethacin that has hydrophobic properties. The systems were studied both with the drug and without the drug, so 12 systems in total were investigated. Each of the system in a wide range of concentration of drug and surfactant. So such selection of model systems cover the wide range of structural properties and micellization and solubilization behavior. So we could assess the applicability of the method to different materials. We started with the characterization of micellar systems. For micelles without API, CMC value was determined using 2 different methods, surface tension measurement and fluorescence spectroscopy. As you can see at transmission electron microscopy images below CMC, there are no micelles in the image. After reaching CMC concentration, we could observe the spherical micelles in the image. After drug loading to micellar solution, the changes in size were monitored with dynamic light scattering and transmission electron microscopy. Before applying differential epitope mapping by saturation transfer difference NMR, we wanted to correlate one-dimensional results, NMR results, to correlate the chemical shift changes with the strength of interaction between API and surfactant. And the other thing, we wanted also to correlate two-dimensional NMR results to correlate set of cross-peaks with quantitative and qualitative composition of the materials. And as you can see, chemical shifts perturbation in one-dimensional spectra indicated changes in local environment of interacting species. Looking at the changes in chemical shifts of the drug substance and the surfactant in the formulation, we could evaluate the part of the structure that is undergoing the major perturbations. In the end, we could evaluate surfactant and drug motifs that were affected mostly by micelle drug interaction. Two-dimensional spectra, in this case NOE nosy spectroscopy, we could see the protons of drug and surfactant that were in spatial proximity. At low surfactant concentration, correlation signals with positive NOE effect were visible, meaning indicating the intramolecular API interactions, and the other correlation signals indicated intramolecular surfactant interactions. With increasing concentration of the surfactant, we could observe additional correlation signals with negative NOE effect that indicated intermolecular API and surfactant interactions. As the result of this set of experiments, we could have map of drug surfactant interactions. And the starting point for a differential epitope mapping by STD NMR is saturation transfer difference NMR spectroscopy. The method has been used so far for host-guest interactions evaluation, and for example, for screening for ligands in case of specific proteins, protein receptors. And in this method, two spectra are registered. The first one is the reference spectrum. The second one is registered after selective saturation of receptor signals. So the irradiation of receptor protons, then the saturation is spread evenly throughout the whole receptor. Then the saturation is transferred to the molecules that are binding. So we are regard them as ligands. So in the end, we observe the different spectrum that is the result of subtracting the two before mentioned spectra. To apply STD method into micelles, it was required to do some assumptions and modification. So first of all, we regard micelle as macromolecules, as the large assembly of molecules. So saturation in this case concerns the surfactant in the micelles. Then this transfer saturation occurs to interacting molecule. In this case, we duplicate the set of experiments. So we saturate either hydrophobic motif and both hydrophobic and hydrophilic motif. At this stage, all existing interactions between API and surfactant are visible, but only the use of differential epitope mapping by STD-NMR gives us preferential localization of API in micelle. And the base of the deep STD-NMR method is the deep STD factor. So it determines the pattern of interaction between two interacting species. Using an example of twin-20 and fluconazole here, we can see that the protons of the drug E and A are in proximity to hydrophobic motif of the surfactant, while the protons C and B are in proximity to hydrophilic motif of the surfactant. In this case, they have respectively positive and negative sign. And I will present the calculation in a few next slide. So the first step to calculate this factor is to register the fractional STD response after irradiating both motifs of the surfactant. Then we need to sum all the values, and the experiment in which the sum is larger will be put in the numerator. Then we calculate the quotient of both experiments, and we calculate the mean value of these quotients. And in the next step, we can subtract the quotient and the mean, which gives us the STD factor, which give us the pattern of the interactions occurring between the respective drug and both motifs. As you can see, applying the method to many samples differing from each other with the concentration of the drug and surfactant give very similar results. The results of the interaction map are quite consistent. Based on these results, we could determine the factors that enable the use of differential epitope mapping by STD NMR method. Only for SLS system, the factor couldn't be calculated. Probably the pool of surfactant protons available for saturation was insufficient in this case. The other factor was molecular weight of the surfactant. So the largest in this case, pluronic, and followed by twin 80, twin 20 at SLS. So the larger molecular weight of the surfactant, the lower concentration was needed to detect the saturation transfer. So it means that aggregate resembled macromolecule to the larger extent, even at low concentrations. Formulation. Another thing is that formulation for which deep STD factor could be calculated were characterized by a ratio surfactant to API extensively exceeding the value required for ligand protein STD measurements that is described in literature. However, after taking into account the aggregation number, which is the number of surfactant monomers that create micelle at specific surfactant concentration, the ratio micelle to API equals the desired value. If we apply the method to several samples that differ from each other with concentration of the drug and the surfactant, the possible drug partitioning between the micellar and aqueous phase could be observed. As you can see here, the solubility within the surfactant solution was much larger in case of hydrophobic molecule, indomethacin in this case, in comparison to fluconazole solubility within micellar solutions. The square points that you can see on the graphs represent formulations that were subjected to STD NMR measurements, and the gray ones indicate the samples in which the fractional saturation STD response was lower than 1%, and the green one represents the samples in which fractional response was higher than 1%. And as visible in the graphs, the STD response was present at much lower concentration in case of a hydrophobic drug when compared to the system with more hydrophilic drug. Overall, the fractional response in the series with indomethacin had much larger values than the series with more hydrophilic drug, which might indicate that the hydrophilic drug in this case has a smaller affinity to micelles that is represented by smaller fractional STD response. In the next step, we correlated the results from deep STD method with other results obtained by other NMR methods. And here is the example for looking at the pattern of the changes in chemical shifts. We can correlate it with the deep STD factor, meaning that the same protons that gave the result that are in proximity to the specific domain of the surfactant, they were undergoing similar changes, perturbation observed in one-dimensional spectra. In the further step, the structure of micelles and the interactions between API and the surfactant were modeled to verify experimental data with computational methods. And in micellar systems without API, it could be observed that the simulated micelles of fluoronic and twin had both motifs exposed to the surface. And in case of SLS system, a typical core shell micelles model was observed with disordered edges of the formed disk. After addition of drug molecules, the surfactant organization in micelles wasn't changed, and the interactions were quite consistent with the STD factor. For example, the fluoro and the chlorophenyl ring of both drug molecules were buried into hydrophobic domain in simulation, and the same results was observed analyzing the STD factors. The interactions at the surface mainly allow to explain the possible use of this method to micellar systems, as the condition for use of such method is the presence of transient interactions between interacting species. To conclude this project, the distribution of the drug between micellar and aqueous phases correlated with changes in one-dimensional chemical shifts. The resonance signals of the less polar API exhibited greater shifts compared to those of the more polar API, similarly to the signals of the surfactants. In two-dimensional NMR spectra, the limited solubility of the API in formulations containing surfactants at concentration close to CMC was manifested solely by API and surfactant correlation signals. As the surfactant concentration increased, intermolecular API surfactant correlation signals with negative Noye effect appeared, similarly to Hosey results. The pattern of intramolecular API correlation signals varied depending on the polarity of the API, indicating a preferred localization in macromolecular aggregates or in the aqueous phase. The innovative application of deep STD NMR method in the analysis of colloidal systems made it possible to obtain a map of preferential API surfactant interactions and information regarding the distribution of the API between both phases. The following factors have been identified as determining the effective application of the method in colloidal materials, such as water solubility and polarity of the API, and the structure, molecular weight, and solubilizing properties of the surfactant. Molecular dynamic simulation have shown that in case of Turonic and twin-20, supramolecular system had a surface composed of both hydrophilic and hydrophobic domains, and interactions with model APIs occur mainly at the micelle surface. In case of SLS system, molecules form classic micelle model with hydrophobic core and hydrophilic surface layer. The second project I would like to present concern the influence of the components of drug-loaded nanoemulsion for ophthalmic use over time. In this case, the alternation in one-dimensional spectra occurring upon storage of the selected formulation were observed and analyzed. Nanoemulsion have many advantages similarly to micelles as drug delivery system. They can enhance the solubility of poorly water soluble API. They might increase bioavailability and biocompatibility. They can modify the release profile of the drug, and they might protect the drug from undesired destruction. In addition, the ophthalmic nanoemulsion have several additional requirements, such as they should have the droplet size below 100 nanometers. They should be characterized by low polydispersity index. They should be stable over during the storage. They should also exhibit extended contact time with cornea, and of course they should be well tolerated. And another requirements that enable the nanoemulsion to be well tolerated are, for example, pH values, osmolality, surface tension, and viscosity. So nanoemulsion might be prepared by low and high energy method. We were interested in formulation with antifungal drugs because such formulation are not registered as marketed products. And in the literature, nanoemulsion with drugs from azole group, antifungal drugs, were prepared only by low energy method. That's why we decided to introduce high energy method, one of the high energy method, which is high pressure homogenization, to get all the benefits that are combined with use of this method. So using HPH, we can introduce large content of dispersed phase, even up to 40%. There is possibility to for emulsification of high viscosity, high molecular weight oils, with at the same time with small quantity of surfactants, and the scalability, the potential of scalability is very large. As mentioned before, nanoemulsion are consisted of two phases, oil and aqueous phase. In oil phase, we can dissolve lipophilic API. In aqueous phase, we can dissolve hydrophilic API with surfactants, co-surfactants, preservatives, osmotic pressure and viscosity modifiers, humectants, gelling agents, and buffers. Upon mixing, the initial emulsification is occurring, resulting in macroemulsion, which is called pre-emulsion. Then this pre-emulsion is put into the reservoir of the homogenizer, and then it is transferred from the feeder to homogenization block. In this block, the material is squeezed through narrow gap under high pressure, resulting in obtaining nanoemulsion. In this type of drug formulation, composition is crucial. So first of all, this formulation are prone to phase separation. That's why we need to have sufficient surfactant and co-surfactant content. At the same time, we cannot add very high content of surfactant because it will cause eye irritation and the leakage of the formulation from the conjunctival sac. At the same time, we need a sufficient amount of oil phase that will be enough suitable for API solubilization. So we did the screening of different compositions. We chose 4 different surfactants: colifor, twin-20, twin-80, and pluronic, and 2 different, actually 3 different co-surfactants: polyethylene glycol, propylene glycol, and triacetin. And the content of each of the phase was in the range between 13 and 20% in case of oil phase, between 5 to 10% in case of surfactants, and between 2 to 10 in case of co-surfactants. So we had 2 series of nanoemulsions. One was with loaded with 0.3% of fluconazole, and another one with 1.0% clotrimazole. The next step in preparing such formulation is optimizing processing parameters. That is why we first try the different pressure values, mainly 500, 1000 and bars and 1.5 kilobars. In each case, the size and the polydispersity index was examined by dynamic light scattering. And based on this screening experiments, 1000 bar pressure was chosen as the most suitable. In the next step, the number of cycles that we used during the technological process was adjusted, so up to 10 cycles. Again, after every passage, the size and polydispersity index were checked by dynamic light scattering, and the optimal results were obtained when 3 cycles were applied. After preparation of the formulation, the nanoemulsions were characterized, and the as you can see, the physicochemical properties of the prepared for nanoemulsions were in the range of the required values, meaning, for example, diameter of the droplet, oil droplets, were around 100 nanometer. The polydispersity index was very low, meaning that the nanoemulsions were homogeneous and monomodal distribution. They had monomodal distribution. The other thing is zeta potential. Those values ranging from around 11 to 30 they ensures long-term stability, and the additional properties that are required for ophthalmic nanoemulsions, such as pH, osmolality, viscosity, and surface tension, were also in the range, in the desired range of values. And the characterization of the nanoemulsions was repeated after stability tests to evaluate the compositions in terms of maintaining their initial properties. And in the literature, NMR was used in nanoemulsion so far to establish drug localization within the formulation. So here we decided to use NMR in terms of stability of the formulation. We wanted to correlate the changes occurring at the molecular level with physicochemical properties after storage of the prepared formulations. So the indicators that were chosen as the indicators of the stability of the nanoemulsions was line shape of the peaks, full width at half maximum, FWHM, and integral values. We monitored drug signals, the OI signals, and surfactant and co-surfactant signals. We compared several series, so those just after preparation, those after, for example, 30 days of stability test or 9 months of stability test. As the control samples, also blank series without the drug were examined, and the drug oil solution also as a control experiment. We assumed that widening of the peaks will be caused by the aggregation process of the droplets. And for example, decreased intensity might indicate that drug is not stable. So it might, for example, sedimenting from the formulation. Here is in fluconazole series, there is an example of the results. Comparing the line shape of the peaks at the NMR spectra, we can observe that twin 80 and low content of propylene polyethylene glycol co-surfactant were they were characterized by the smallest stabilizing effect. Another thing, when we consider FWHM values, for example, in series of nanoemulsion with colifor and different content of polyethylene glycol, we also observed that a small amount of polyethylene glycol resulted in less stable nanoemulsion. When the same was applied to colifor series with other co-surfactant, propylene glycol, those changes were not observed, which give us the information that propylene glycol is better stabilizer than polyethylene glycol. Another example is the twin 80 series with propylene glycol as co-surfactant. In this case, we can see that lower stabilizing features of propylene glycol are visible at 5% when compared to 10% content in the formulation. Looking at the drug signals, we observed that drug is most stable in colifor nanoemulsions with propylene glycol and polyethylene glycol at higher concentration. In other cases, we observed both the increase of FWHM and decrease of intensity values. Similar observations were analyzed in clotrimazole series. However, FWHM values were unchanged when the series after 30 days and freshly prepared compared, which means that this kind of nanoemulsion were much more stable than fluconazole series. It might be a result of different oil phase content in both series. So another examples when considering FWHM values in twin 80 series, they were much increased when triacetine or propylene glycol as co-surfactant were used, and it was also correlated with the increase in size of these nanoemulsions examined by dynamic light scattering. Another example is also when considering FWHM values in nanoemulsions that included triacetine as co-surfactant. Regardless the surfactant used, they were much increased, and it was also correlated, for example, with large change in zeta potential value, meaning less stability. Looking at the drug signals in the clotrimazole series, we can observe that line shape changed in all twin 80 nanoemulsions and coliform with triacetine, and in this case it was correlated with the largest drug content decreased established by high pressure liquid chromatography. To conclude, NMR spectroscopy was used to follow the occurrence of alternation in nanoemulsions at the molecular level upon storage. The changes regarding the line shape, the integral values, and the full width at half maximum of the NMR peaks corresponding to either the oil or surfactant phase or drug molecules were evaluated as indicators of the nanoemulsion stability. The widening and the lowering intensity of the peaks might indicate the aggregation process between the respective components. The higher ratio of FWHM between samples after the stability test and freshly prepared samples characterized the composition with lower stability. NMR data confirmed the results obtained by other analytical techniques, namely the coliform-based formulations provided higher stability in the nanoemulsion composition compared to the use of twin 80. Moreover, a better stabilizing effect of propylene glycol as a co-surfactant was observed when compared to polyethylene glycol. Triacetine had the lowest stabilizing properties among the considered co-surfactants. I'd like to acknowledge co-workers that were participating in the projects about micelles and nanoemulsions from Wroclaw Medical University and University of East Anglia. And also I'd like to acknowledge the funding of the projects about micelles and nanoemulsions that were mainly obtained from Wroclaw Medical University and National Science Center in Poland. Thank you very much, and thank you for your kind attention.
Thank you very much, Katarzyna, for a great talk. And we are open for discussion. If you want to ask questions, please turn on your microphone and camera. I especially encourage students to ask questions as we are still competing for Best Student Question Award. Do we have any questions? Oh, I think Krzysztof has.
Yes, I have a question. I'm not a student, but I have a question, and I have a very basic question. So perhaps it could be asked by a student as well. So that's a question about CMC and in vivo applications. Because according to my understanding, when you use micelles in a drug formulation, they got diluted in the bloodstream, for example, right?
Right.
And then you fall below CMC, or not?
Yeah, that's correct. That's why our idea of preparation missiles was the application, topical application, for example, into the eye, as the eye drops or eye sprays, to avoid this solution. Because you're right, getting below the CMC, it disintegrate. They disintegrate into separate monomer.
Okay. So that's the real problem, right?
Yes.
Okay, and how about other factors that occur in vivo, like, for example, pH or temperature a bit higher than room temperature that you used?
Again, it depends on the route of administration that you choose. Some of the experiments we were also performing at higher temperatures. I didn't show the results here, but we also performed the experiments at 37 degree temperature. That should be the body temperature approximately. And all the pattern was quite similar in this case.
Okay, thank you.
Thank you.
Do we have any other questions? Let's give it a moment. Okay. Students are very shy this year, apparently. Okay, so if there are no more questions, I'll just show the last slide of today. So this was the very last seminar of this semester. So have a great summer, and I'm working on the schedule for autumn semester. So if you have some interesting speakers that you would like to recommend, please send me the email. I would gladly use those suggestions. So thank you very much. Thank you both our speakers, and see you in autumn.
Thank you. Goodbye.